Okay, thank you. And I would like to first thank the organizers for this opportunity. And I'm really glad to be here as an ex-UEA employee. I stayed in Norwich for 9.5 years, and I really had really great memory in Norwich. I did actually swim and run and drink in UEA, and it's a great moment. So, and also I'm very happy to be here because I'm learning a lot of our history in Japan. So that's a great opportunity for me. Okay, so today I'd like to talk about parasitic plants, especially Striga. Oops, already something going on. This is my Mac, but somehow missing. Sorry about this, but I want to say just that there are so many parasitic plants in the world. So it sounds weird, but it's actually quite common. In fact, the plants evolve 12 times independently to become parasites. So it's kind of common way of living. And you may have seen that. Okay, so the mistletoes or scuta or the largest plants in the world is also the parasitic plants. And we are studying this family called Orbaceae, or commonly known as a broom rape. And these broom rapes are everywhere in the world. And actually you can see in the UK. This is the photo I took about 2 years ago when I did the sabbatical in Cambridge. And this is, the building is the new Sainsbury lab in Cambridge, and just nearby the tons of Orbaceae you can find. And also in Japan, if you like hiking in the mountains, you may see a lot of Orbaceae plants in Japan. They're many, pretty nice. And these flowers around like August and September time, so if you have some time, you can just go around and see. And one of my favorite ones is the Pterosphaerum, and I talk about these plants later on. And so this, somehow the Japanese are fascinated by this Orbaceae plants, and it's actually history back to the 8th century. So you can find actually the poem in this famous 10,000 Leaves collection. It's one poet wrote about this flower. I can read in Japanese. 道の辺のお花が下の思い草、今さらさに何をこの. So this is the poem he found on the way, that these plants, very weird plants, facing down, and then this thinking something, and that's actually similar to him. He is thinking about somebody else, and he's like really looking like these plants. And this poem is actually very unique because the poem series is usually featuring one plant in PowerPoint, but actually they show the two plants. Here's one is Obana, here's this pampas grass or miscanthus, and also this is the parasitic plants, Omoius, right? Because this is actually the host of this plant. That's why they show this too. So I think this is probably the first sort of description of parasitic plants at least in Japanese history. Okay, so this is Japan way of thinking about, but this is Chinese of thinking about. So Chinese, of course, may like to make a medicine from plants, weird plants, and this is the plants found in Taklamakan Desert. It's almost endangered species because they produce a very good medicine. And in fact, Japanese use these drugs to create this super energy drinks. And this, you can see this convenient, you can really find this sort of the bottles of these energy drinks. And one of the most expensive ones actually contain these particular drugs from this plant. So that's one of the reasons they are become an endangered species. So, anyway, so the Orbaceae plants, most of the Orbaceae plants are parasitic plants, except one called Rindenberger. This is a non-parasitic plant. So something happening in evolution that this become a parasite, become lazy, and some actually even lost the photosynthesis, and some actually become obligate, so you need a host to get all the nutrients and become the parasite. And in fact, many of those Orobanche plants actually have big problem. And especially the Orobanche and the Striga are the most devastating parasitic plants in the world, and especially in the Mediterranean area or Africa, northern part of Africa, has a big problem of having these plants. And I can show you some of the plant species, how bad they are. Okay, so this is the carrots infected by this Orobanche crenata in Israel. As you can see, these white flowers are the parasitic plants, and these are the green ones. These are the carrots. Basically, this field is dead, so you cannot grow the carrots anymore again. And same thing here for the sunflowers. This is Orobanche kumara, again in Israel. You see this small plant, these are parasitic plants, and basically these sunflowers are all dead. And this is a tomato infested by the Felipanchi ramosa, and these are the Felipanchis. And again, they cannot grow tomato anymore in this field. So I asked what are they going to do with this field, and they said just build houses instead of doing farming. So this is how bad it is. But especially the bad one is this Striga, or the people call the witch weed, because this looks like very beautiful, but really devastating. So this is the witch. And so this is the photo I actually took in 2010 in Sudan. It's very near to this border of South and North Sudan. There are lots of the sorghum field, especially they're using the Nile River to use the water to irrigate. But these irrigation actually bring all the Striga seeds around, and you can see the Striga flowers, the purple ones. And the farmers of this sorghum field, he said that the previous year was almost none. This was a sorghum field, and this year it's just wiped out. And this field is actually dead. They cannot grow any more of the sorghum. So this is how bad it is. And that you can see this how this the parasite comes. So this is a normal sorghum, but once infected like this, and you see you don't get any grains from this infected plant. So it's almost 100% affected. So this is one reason why this was listed. This is seven bad guys to threat food security, and as you see in cereal stock. And the Striga is actually making about $1 to $7 billion loss per year, which is very similar to the other really infamous pathogens. Okay, so these guys are bad. So why so disruptive if the plants, people can just go out and pick it up? But the problem, the secret is in the seeds. So this is the size of the seeds I can show. This is a corn, as you can see, this is a sorghum, and this is a Striga. Striga is almost this spot, it's almost a dust. So they don't produce any of those big sort of starches. So they make very, very small, tiny seeds and they spread like crazy. And each plant make 100,000 seeds. So this is the big problem. And these seeds are really tough, and there's no problem in 30, 40 years they stay, and they're strong in extreme dryness. So such as like a Sahara desert. And even no germination, even the water coming by flood or water, they don't germinate, and they germinate only next to the host plants. So that's the trick they do. So about 50 or 60 years ago, that actually the compound which induces this germination as isolated. It's called it Striga because it's inducing in germination of Striga. And this Striga is very unstable, and it breaks down very quickly in soil. That's why within millimeters, they can detect the host, and then only the host is nearby, they can infect, and they can grow only this size in the first day or so. And if they couldn't find the host, they die. So in this way, they don't really need a starch to build the actual roots, right? They just need millimeter and then they can infect. In this way, they can spread seeds like crazy. Okay, so it's a big problem. So one way is to fight against this striga is actually stop this strigal production from the host. And people wonder for the last 60 years why plants has to produce this particular compounds to actually attract the parasite. And the reason why they do this is actually striga is released into the rhizosphere not to attract the parasite, but actually the symbiotic fungi. So the symbiotic fungi actually goes attracted by these chemicals, and it goes into the roots. So when the host needs a phosphate, they produce this strigo to attract the fungi, but parasitic plants actually hijacking it. Okay, so the one way is actually to make a resistance is actually find the strigal biosynthesis pathway and block it and see what happens. And this is the actually experiment from the paper we published. And sorry, this is only the experiments I show this time. It's a rather busy slide, but what I want to say is this is D10 is a biosynthesis mutant. Is in D10, and this is a disease scores, and you don't see any disease in this particular mutants. So this means that the striga is not really infecting this particular biosynthesis mutant of rice. So you could do that. And in fact, if you're looking at the striga side, so we sequence the striga genome and see what sort of strigolactone receptors they have. And in fact, it's actually, they have huge numbers of strigolactone receptors. So in Arabidopsis, like model plants, it's just only one type of this strigolactone receptor. But the striga has more than 20 of them. And they are probably slightly different, the reception site. So they can actually detect many different kinds of strigolactones. And they probably can enlarge the host range. So what we need to do is that you don't have to really block the strigolactone biosynthesis pathway completely, but it can change the strigolactone structure so that it still works for the attracting fungi, but not attracting the striga itself, right? So, and then you should actually learn from this the structure part of this receptor and then see whether that is possible. And in fact, this month, the American groups from Purdue, Dr. Ajeta's group actually published a paper in PNS that the particular mutant in sorghum actually has this mutation which actually combats strigolactones in a different way. So they produce different types of strigolactones, and they have become complete resistant to striga. And you can see this normal type is the striga infested like crazy, and here is very resistant. And you can actually see this is a little bit of stunted. So maybe this particular compound is important for the height of the sorghum, but anyway, this plant become resistant to the striga. So you can actually use this kind of knowledge to create this resistance against striga. So other way of making striga resistance, as Cyril sort of mentioned a little bit, is finding the receptor against striga. And this paper published in 2009 identified the internal receptor for striga in cowpea. So in cowpea and striga interaction is a little bit different from sorghum striga interaction. The cowpea has some particular race or cultivar species that actually is very strong to striga. And this Michael Timko's group in USA, they did the actual cloning of this particular gene from the cowpea. And this is actually called the R gene, which encodes NBS LRR type intercellular receptor. So this kind of receptor genes can maybe transfer to these crops and then see to see the resistance against striga. That's one possible way. Okay, so I just like to show you some striga infection as an interesting biology. So here's the striga, see this strigolactone from the host, and then germinate it, and a few millimeters away from the host. And then the roots actually somehow fan and then goes towards the host and then start differentiating in making it like a callus. It's like really like a tumor-like organ just came out and then attached to it and then start invading the host plants. And this is the striga infecting the corn roots. And when you cut here, you see this by the microscope, you can see this is the striga, and they actually connecting the vasculature of its own to the host, and then getting all these nutrients and water. And this particular organ, it's like a callus. It's like a cancer-like organ. It's called hostology, as a special word called like invading organ. Okay, and we know now that the particular organ called hostology is induced by this particular compound called DMBQ. So this is lignin-derived quinone. And now we clone this genes encoding the candidate receptor for this DMBQ. So we may able to later on that fight against these plants by using this kind of receptor. Okay, so apart from water and nutrients, and any other materials exchange, because their interaction is quite intimate. And how about genes? Any genes are actually transfer each other. And in fact, we found that this particular gene, we call it 9483, is actually coming from sorghum and then gets into the striga. So this is found in the striga genome and also the sorghum gene, very similar each other. So if you make a tree of this cluster gene, so it be clustered together with the sorghum. And this gene is monocot specific gene. And you never find in dicot plants. But in fact, the striga is a dicot plant. So dicot plants somehow get the gene from monocot plants, and it's most likely the sorghum because this is the closest one we found. So what happened? So what this means is actually when it's interacting, this gene is moving into the striga. And how? Because this gets into the striga genome already. So that means this has to go to the flower and then goes into the seed and goes to the next generation, right, somehow. So maybe the chance of getting into this other genome must be very, very low. And we think actually this gene transfer occurring very, very often, only this one somehow managed into the seeds. Okay? How about other? So when we checked this striga genome, we found many retrotransposons, actually specific to the sorghum. This is sorghum retrotransposon somehow jump into also the striga gene. So genes or retrotransposons is probably messenger RNA. Maybe the last case, this case is also maybe messenger RNA actually move into from the host to the parasite and gets into. So lots of probably information exchange maybe occurring from between these two species. Okay, so if you want to study really more about this intimacy between these two plants, we need a model because the striga it's relatively difficult plants to work on, and that's why we find a model. And I chose this Pterosphaerum japonica because this is Japanese native, and it's short life cycle, and genome size is okay, and it's a selfing plant, so it's easy to grow, and also this plant doesn't require any host to grow, so we can grow this plant very easily. So in case of a striga needs a host, so you always have to play with the host and parasite. It's difficult. So, and these plants can infect many plant species, including Arabidopsis. So, and I trained Norwich-born British-Japanese scientist. She was born in Norwich Hospital, and now she's in Japan, and they're looking for this P. japonica, Pterosphaerum japonica. In Japanese, by the way, it's a Koshogama. And now she can spot better than me, and we collected several lines of this P. japonica. Anyway, so I can show you another videos that this is the Sorghum P. japonicum try to infect this model plant Arabidopsis. But this case is a little bit different from Striga. Oops. Sorry, I couldn't show it, but it just has to go and then infect. I didn't really check this system, I'm sorry, but believe me, this goes and infect. Okay, okay. So one another interesting thing is this P. japonica actually make host fat, as you can see in this photo. So this is the infection site, and this is Arabidopsis root, and this upper part is much fatter than original Arabidopsis root. So by injecting something and make the host fatter for themselves. Right? And what we found is actually this parasitic plant injecting growth hormone of its own. It's called in plant cytokine. Producing in parasite and they inject into the host. So here, this is the marker in both the parasite and the host. And you see this, when the cytokine is produced, you can see this purple. And then because it's injecting into the upper part of the host, you see this green color as a sign. And we have also genetic and biochemical data set to prove this is the case. So they are really doing intimate interactions, even by injecting hormones. Okay, so we continue this work. We have this massive P. japonicum field in our lab and mutagenized. And we can get the mutants. So some are really, this is a wild type, and some mutants you don't get any hosterium or elongated hosterium or no hairs. And now, because of this next generation sequencing technology, we can actually clone genes very quickly. And now we have 5 new genes that are controlling this effect. So now we are studying this. So one day, this will tell you how we fight against Striga, and maybe Japanese parasitic plants may help this. Okay, so summarize my talk, and we think resistance to Striga can be achieved by studying the plant-plant interaction in molecular level, and we think plant-plant interactions are intimate, and studying such interaction would reveal new biology in a much broader sense, such as the Striga biology. And here, just in the last slide, I'd like to show the plant immunity research group in RIKEN, and I circled the red with the red pen that the 4 people actually came from the Saint lab. So 2 Japanese, 1 French, 1 German, and also I have 2 persons from the Cambridge University. So we have quite tight connection with the UK science. And I'm sending 2 students, Ayako and Yuki, to Sainsbury this summertime. So we continue to work on together. And I stop here. Any questions? You are. Thank you very much.